Thank you, Joanna, for sharing your family story with us. That was such a wonderful example of the importance of the need for drug development for Duchenne's muscular dystrophy, and also for the important role that data and studies play in the process of developing these drugs. I also want to thank the speakers who've gone before me, Justin, for giving the example earlier on Duchenne's data, because I think that'll make it really accessible as we go through this next session, where I will discuss the benefits of integrating multiple data sets for rare disease using Duchenne muscular dystrophy as a case study. So first, we're just going to start off with following up a little bit on the theme that was just discussed in the immediate presentation, connecting the dots with rare disease data. So this is just an example of transcriptomic data looking at some inflammation genes in patients who have Duchenne muscular dystrophy and those who do not have Duchenne muscular dystrophy. In this network diagram, the genes that are increased in the subjects who have the disease are shaded in red or warm colors, and the genes that are lower are shaded in blue or cool colors. And this is just an example of how there is extreme connectivity and a lot of network within, in this case, these are inflammation genes in Duchenne. This particular study was looking at these changes in inflammation genes and showed that this was a relatively early occurrence in the disease. But what I wanted to illustrate with this is that there's a lot of data and a lot of dots that need to be connected, and that we can actually put some resolution to the image of what this data can tell us if we integrate these disparate data sets into other data sets to enhance the analytical power. So this is just a quick outline of what we'll cover in the next couple of minutes. I'm going to talk about Duchenne muscular dystrophy, that I will abbreviate as DMD for simplicity, just a quick background. We'll talk about the clinical trial design landscape in DMD. I'll introduce the Duchenne Regulatory Science Consortium and the mission of the Duchenne Regulatory Science Consortium, which I will abbreviate D-RSC. And we'll discuss the data sets within this consortium and how we've integrated these data sets and the data from them into a really exciting and highly functional data set for DMD. We'll wrap up with the regulatory pathway and where we are in developing these tools using these Duchenne data sets for regulatory approval. And then we'll end with some future directions for the D-RSC consortium. So this is just a schematic to give a little bit more background. Again, I'm so thankful that Justin provided some really useful background on Duchenne muscular dystrophy. It is a rare genetic neuromuscular disorder that occurs in males. It is X-linked, affecting between 1 in 3,500 to 5,000 male births. The genetic mutation is in the DMD gene, and this gene produces the protein dystrophin. Dystrophin is required for muscle function, and it is the loss of dystrophin in DMD that leads to the pathology. So this chart illustrates this pathology as it progresses over time. Because the mutation in DMD is present since birth, the initial signs are actually observed as developmental motor delays. And this is observed early in youth, in young children. And it continues to progress from there to functional deficits. This occurs in approximately 8 to 11 years of age. After that, entering into the early teen years, the people with Duchenne's are losing more function and are now non-ambulatory, usually requiring full use of a wheelchair. They also have increasing loss of motor function in their upper limbs. In the more advanced teen years, deficits in respiration and difficulty breathing manifest. Often the people living with DMD require ventilation support in order to address this issue, and they continue to have progressive loss of ability to perform daily functions. In the late teens to early 20s, at the more advanced stage of the disease, there are increasing cardiac dysfunctions, increasing respiratory deficits, and this will eventually lead to premature death. There's no current cure for Duchenne muscular dystrophy. There are a handful of drugs that have been approved. We'll talk about the clinical trial landscape in just a moment. But importantly, there aren't any drugs that can stop the disease or reverse it. And so this is why the drug development in Duchenne's is an active area of progress. There are, however, some challenges to it. I mentioned earlier that the DMD gene produces dystrophin. Turns out that DMD is the largest known gene in humans, and it has a very high spontaneous mutation rate. This makes it a bit of a tricky target. So on the one hand, there's the advantage of DMD being a monogenetic disease, muscle being a tissue that doesn't divide, and therefore approaches to cure it, for example, with gene therapy, seem amenable in the sense that you could get a lot of effect from a single administration. But there are a lot of technical challenges because muscle is a very large organ. You need to be able to target it throughout the body, since the previous slide illustrated that the defects in dystrophin are in all muscles, in cardiac, diaphragm, skeletal muscle, etc. So there are a lot of challenges to the drug development landscape. This particular chart is summarizing a couple of options. I know that the print is a little bit small. I'm not going to dig into the detail here. Just want to provide a conceptual overlay for how normal drug trials are typically very long because it's such a complicated process. And there's a lot of high failure rate and a lot of upstream analysis that needs to occur. And this can take approximately 12 years. But in a rare disease, using a gene therapy approach like Duchenne muscular dystrophy, this clinical trial process can be sped up a bit. And that's simply because there is additional steps that are undertaken in the preclinical stage of the development that allow for the additional testing on safety and dosing and other considerations to be addressed in the preclinical phase before it enters the clinical trial. And then phase 1, which is usually a trial to test safety of the drug approach in healthy subjects. And then phase 2, which is testing the safety and efficacy in disease subjects, where that is separated in a standard clinical trial for a rare disease gene therapy trial that's combined into the same phase. And then last, in phase 3, where the efficacy of the trial is evaluated in a larger group of patients, that still occurs. But I do want to point out that because of the urgency and the need for advanced therapeutics in Duchenne muscular dystrophy and the accelerated approval processes that are available for rare disease, this can occur a little bit faster in a gene therapy approach. So this is a chart that summarizes the clinical trial landscape in DMD. I mentioned that there's no current cure to stop, halt, or reverse the disease. There are a handful of drugs that have been approved. Predominantly, the standard of care is administration of glucocorticoids. There are a couple of other therapies that have been developed, but actually there's regional differences into how these therapies have gone through the regulatory process. For example, there's an approved drug that has been passed through regulatory approval in Europe through the EMA, but that's not available in the US, and vice versa. There's another drug that has passed regulatory approval in the United States, but not yet in Europe. And clearly this highlights the need for continuing the necessary work in drug development in Duchenne muscular dystrophy. And that is ongoing. So the last decade has seen a lot of increase in clinical trials for this disease. And specifically this chart, which is actually by now a couple years old. So there's additional progress that is not summarized on this particular figure. I want to point out that there are a wide variety of drugs that are now in development. And these include approaches to restore or replace dystrophin. So this could either be through gene therapy or through some other approach. There are drugs that target inflammation and fibrosis, calcium regulation, cardiac function, blood flow, mitochondria in order to provide additional energy for the muscles, and also drugs that aim to approach muscle growth and protection. But again, there's urgent need for this process to continue and to be more efficient, because even the few drugs that have been approved for DMD only treat a minority of the patients. And often certain types of drug development can only be applicable to a small percent. And this is already a small percent of a rare disease population. So we do need to enhance our approach and really up our game. these new approaches are absolutely required. I compared earlier some of the examples of progression through a standard drug development procedure for a clinical trial and the many years that takes. We're looking for ways to accelerate this process. So instead of using the traditional approach that relies on limited information, small data sets, potentially outdated data, and a bias to the most recently published paper, we're now looking to more creative options like data and quantitative model-based drug development approaches. These use integrated global data sets, including relevant populations and endpoints, and these allow the development of quantitative models of disease progression and engage the patient population and endpoint behavior in an integrated fashion. So this is just a quick summary of the Duchenne Regulatory Science Consortium, D-RSC. The mission is to accelerate therapeutic progress and establish novel drug development tools to better inform clinical trial protocols for Duchenne muscular dystrophy and reduce the number of patients needed to demonstrate efficacy of new therapies. The data is incredibly important to the D-RSC mission. The data is integrated from multiple different studies. It is then curated and cleaned in some of the ways that we spoke earlier that the platforms need to address. It's then integrated into a database, and from there, models of either disease progression, efficacy of biomarkers, novel patient-reported outcomes. These are just examples of the types of analyses that can be applied to this integrated database. This can also feed forward into a clinical trial simulation tool, which I'll tell you about in just a moment, with, of course, the end game goal being regulatory review and acceptance. So this is a schematic of how this process happens at the more granular level. The data is input from multiple different sources. Again, we really appreciate the contribution of data from all of these different data owners. You can see that this is a combination of registries through patient advocacy organizations and also from biopharmaceutical companies. That is then integrated into a modeling schematic that allows for endpoints at baseline and also analysis over longitudinal components to be modeled. Eventually, the output is, back one. The output is to better understand the disease and its progression, to better understand the trajectory, the rate of progression. I pointed out earlier that there's that continuum, that spectrum of symptoms that happens in Duchenne muscular dystrophy. And while there is a structured order to that progression, there is variability from patient to patient with how fast they progress. So within any particular data set, there can be wide variability with the rate of progression of the patients through the spectrum. And so integrating all of this data, modeling it, and developing this output into a clinical trial simulator can really help us as we move into the clinical trial space. This is an example of how that occurs iteratively in these quantitative models. So the goal, of course, is to take this data that has been integrated, understand what it is telling us, use that to simulate and provide an in silico environment in order to address many of the key questions of a clinical trial, to ask questions about progression, exclusion and inclusion criteria, the frequency of patients having to drop out of a study, for example. All of these deep questions that can affect the success of a clinical trial can be simulated in silico in order to really better understand what considerations and criteria need to be met, and also what outcome assessments are best suited for the clinical trial. Many of the natural history studies span a few years or several years, but the typical clinical trial is maybe only 18 months or 2 years in duration. So there may be certain clinical outcome assessments that are more sensitive to detect changes in the period of time of a clinical trial. All of this, of course, requires sophisticated mathematics and analytics in order to continue the simulation and make the tool more and more meaningful. This is an example of how a clinical trial tool like this can be implemented in a potential real-world example. So one of the things that we are pursuing and something that we're very interested in for Duchenne muscular dystrophy is the concept of a platform trial. This is a clinical trial design that standardizes a master protocol and allows multiple drugs to be evaluated simultaneously. It reduces the number of placebo controls. It increases the efficiency of the clinical trials. It allows more rapid selection and more nimble decision making. And this is an example of how a clinical trial simulation tool could be used in a platform trial in Duchenne. There would be different goals, for example, of to look at for either early outcomes or potential complications. There could be specific goals that are looking at, for example, progression to ventilation. This would be a later stage change after ambulation is lost. And you would be able to use that clinical trial tool to evaluate different candidate endpoints to be able to see which ones are better suited for the particular goal in mind. It will also help enrich and stratify the patients as they're being selected and screened into these different trials. It will also allow for models that are more appropriate to that particular outcome to be applied. So this is just one example of how the clinical trial simulation tool could be utilized in Duchenne muscular dystrophy. And again, we are also working on developing a platform trial and master protocol for that effect. So specifically, how will these models improve the clinical trial landscape? They will develop a mathematical understanding of how the endpoints change over the course of the disease, so assisting in measuring progression. They will help increase understanding of the sources of variability in the rate of change. This will help understand how to properly power and how many patients are required, may be able to reduce the number of subjects who are enrolled. They will improve the comprehension of how changes in one endpoint are associated with the changes in the later stages of the disease. We heard earlier of how important it is to measure something clinically meaningful and something that matters to the patient. So the clinical trial simulation tools can help us identify that. And these can also be used to map disease progression to changes in these meaningful outcomes. For example, specific to DMD, loss of ambulation, loss of the ability to transfer from chair to bed, moving on and progressing to ventilation, etc. It's important for these clinical trial simulation tools to be regulatory approved. It's important for many different reasons, one of which, of course, is that if it has been regulatory endorsed, and what I mean by that is that regulators agree that the drug development tool is appropriate for a specific context of use. Once it's approved for that context of use, then other drug sponsors who are pursuing a similar context of use can use the tool because it's already been approved, it's already been vetted by the community, and it doesn't have to be entertained on a case-by-case basis, which would slow down the clinical trial. This is very important because it increases confidence in the tool. It makes the clinical trial development faster, more efficient. And this enhances and benefits the patients because, of course, they're able to see progress more quickly, potentially reduce the number of placebo subjects within the trial. It benefits the research community because it makes the outcomes much more understandable and the variability more easy to grasp and limit. And it also benefits drug developers. So this is just a quick summary. On the left, we're looking at one particular mechanism for how these drug development tools can matriculate through the FDA, through the particular fit for purpose pathway. And then there's a similar parallel process through the EMA called the quantification of novel methodologies. Both of these regulatory mechanisms are designed to evaluate these drug development tools, like the clinical trial simulator that I mentioned earlier. And there are multiple steps in the process to confirm that it is rigorous and has the confidence to be included in clinical trials moving forward. So within the Duchenne Regulatory Science Consortium, we're working on this clinical trial simulation tool, integrating these multiple data sets with the aim of applying them through both of these pathways to get them approved globally. So just want to wrap up with some future directions for the D-RSC consortium. I mentioned the clinical trial simulation tool. We are continuing also to develop additional disease progression models and to explore other multivariate models. All of these, of course, require integrated data sets. We're working on developing models of fat fraction in muscle as imaged by MRI. This is a different clinical trial simulation model that is under development. We, of course, are working to get those through the regulatory pathways. Other work within the consortium is looking at qualification of glutamate dehydrogenase as a marker for safety with a liver panel in clinical trials. We're also developing that master protocol for a platform trial. We're looking into a lifetime mobility scale for Duchenne muscular dystrophy. Of course, we're working on continuing to expand our database, which currently sits at 24 data sets. But because it's a rare disease, we need more data in order to enable more powerful analytics. And we are working hard to bring in cardiac data, Becker muscular dystrophy data, and fluid biomarker data into this ecosystem. So with that, I just want to take us to the end where we have a moment to have a panel and engage a few question and answers. I specifically want to welcome a partner and a member of the Duchenne Regulatory Science Consortium, Joanne Donovan, who is a clinical medical officer at Edgewise Therapeutics. And Edgewise is a biopharmaceutical company that aims to develop novel therapeutics for rare neuromuscular disease. Thank you for joining us, Joanne. Let's see what questions we have.
Hi there. That was a very nice overview there. And one of the things that I would say is it's so important for pharmaceutical companies to have this on the horizon of something to do at the end of a trial, because there is so much going on at that point, particularly if a product does not meet its primary endpoint. That should be one of the highest things on the priority. We've heard so much today about how important it is to provide patient data back to the community. And that is something that should be very high on the priority list in terms of wrapping up studies. And so it's important to get it back and to contribute these data sets. So.
Absolutely. And I think that that's a very important key. In fact, we've heard earlier from other people on the panel and discussions about sometimes a resistance for people to share the data. But you, at a company that is looking at in the rare disease space, can you just help us understand at what stage of your drug development process do you need to have some information about this data? Is it early, middle, late, all of the above?
Yeah, I think it's very important early. You mentioned, for example, when you are developing clinical protocols, how do you decide what patients are going to be included or excluded? And a lot of times in that process, people have a lot of great ideas about, okay, we'll include this set of patients, we'll include that set of patients. And then, but if you're actually looking at real data, you can see what that does to the total number of patients. And you may end up with conflicting inclusion criteria that you end up not being able to enroll patients. And again, that's delaying things. And we all know that the urgency is there in this field to be able to move things ahead. So it's very important from that point of view. It's very important in terms of variability and understanding, because it's all about signal to noise. So if you can understand where variability is coming from, then that helps you also improve, understand your sample sizes you need better. So
perfect, exactly. And
those are very important parts.
I agree, and I think it's a conserved trend that the whole field that's developing drugs for DMD is approaching. We also had a question about whether or not the DMD data is available through the RDCA-DAP platform, if this will help advance progress for the disease and encourage and how we're encouraging DMD scientists to present the data. So I can take that one. We have some data sets that are available through the RDCA-DAP platform. With some of the DMD data that we have within our D-RSC database, as Justin alluded to earlier, some of it is just not available to public users because it's restricted to internal use at the consortia. But we are working to bring all of our new data sets in at a higher level of shareability and integrate them into the RDCA-DAP platform so that they can have more utility. I think we have time for one quick question. We're actually running a little bit over, so of course we'll have to get to some of these questions offline because we did get a lot of great questions into this panel. We have a question about the difference, the distinction between a regulatory review of a quantitative solution versus the regulatory review of an endpoint. And I think that's a very important distinction, because there can be regulatory review clearly for things like biomarkers or specific clinical outcomes. But what we're talking about in this unique circumstance of the clinical trial simulation tool, we're integrating a lot of the data into a machine learning algorithm tool that is then considered its own entity within the fit for purpose or EMA pathway. So it is a distinction that needs to be understood when you're going through the regulatory process. But I think now that we are actually coming up on a break, I want to thank you, Joanne, for joining us on this panel. Of course, for all of the advisory benefit that we get from you, having you on our Duchenne Regulatory Science Consortium, and thank everyone for their attention to this very important topic. We'll go to the break. I think we'll reconvene at 2:10 p.m.